Hello, everyone. My name is Zane Thornberg. I'm also a graduate student in the Luthi Schulten Lab, and I'm going to talk about combining some of the work that both David and Ben have talked about in our current submitted publication, Determining the Rules of Life of a Minimal Cell, specifically of Syn3A, and that is currently in review. So all of the results today are in that. So first of all, there's been a lot of talk of stochastic simulations today by my coworkers, and the way that we do this for chemical reactions is through something called the reaction diffusion master equation, which describes the time evolution of the probability to be in a state X, where the state X contains the coordinates and the chemical identities for each particle or molecule in your system. And so, as Ben showed, we discretize space to a cubic lattice, and among that lattice, we allow particles to diffuse among neighboring lattice sites with this diffusion operator. And then once two particles are within the same lattice site, we allow them to react. So each cubic lattice site is considered well stirred, and therefore particles are allowed to stochastically react according to propensities or rate constants from the vector called A, and then determined by their stoichiometry, which is here S. And you'll notice next to this reaction operator, I also wrote the chemical master equation. So you can also remove the spatial resolution of your simulations entirely and assume that your whole system is well stirred. So getting rid of that sum over all subvolumes V, you just have the sum over all reactions. And effectively, you get a system where everything is allowed to react, and you assume that nothing is diffusion limited. We have performed whole cell simulations of Syn3A under both of these assumptions, both a spatially resolved model and a chemical master equation well-stirred model. And so to expand a little bit more on the algorithm that David discussed earlier of our hybrid methods, I expanded this along with some code of a previous postdoc left behind to expand it to including a reaction diffusion master equation step where we do this same method where we perform 1 second of simulation time, only now there's an initial step of doing the top level is now doing a spatially resolved simulation, halting it, updating particle counts into a well-stirred model of the cell, so a global CME assuming the whole cell is well-stirred, perform that second of simulation, halt that one, and then communicate costs of proteins, nucleotide costs, and then protein counts to the metabolism, and simulate 1 second of the metabolism using ODEs, and then you have determined reaction fluxes. And then the resulting metabolite concentrations are communicated back to our stochastic reactions to influence the reaction rates. And so to construct a whole cell model in the spatially resolved sense, Ben's already gotten you through the cryo-electron tomograms, which is the panel A here, and then we place the chromosome configuration. My configuration is colored the same as his. You can see my coloring scheme here. And then the orange is me removing the coloring scheme just to show that it is the chromosome. But in C, you can see that there's a green cubic lattice that represents the membrane. So I've put the ribosomes and the DNA within a membrane. In D, we start placing some membrane-bound particles. So the red are the bacterial degradasomes, the complex that breaks down messenger RNA, and the light green are the sugar transporters PTSG. And then in E, here we have placed the 77,000 proteins from the entire proteomics of Syn3A into the membrane and cytoplasm. And as you can see, it's quite a crowded organism once you put everything in there that should be. And so we will do 20 seconds of simulation time of the spatially resolved model. As Ben said, we keep the ribosomes and DNA configurations static in this at the moment and are focusing on the reactions. But every time a ribosome goes from yellow to purple, it is actively translating a messenger. Whenever a degradasome goes from red to blue, it's actively breaking down a messenger. And whenever you see a green dot on the orange DNA, that's an RNA polymerase reading that gene. And so what you see here is sped up 20 times. It's 20 minutes sped up to 1 minute of simulation time. And you'll notice that the DNA configuration is actually very critical here in terms of determining kinetics, because if a gene is more farther into a cell, then it's going to take it a longer time for it to reach the membrane-bound degradazomes. So the mRNA half-lives in our simulations, the lifetimes of these are dramatically affected, and also accessibility to ribosomes. And so that's why in our simulations that we presented in the paper, we ensured that each configuration used among each replicate cell was varied from the others in terms of how deep into the cell the average gene would be. And also noteworthy is that in this simulation, we do the transcription of all 493 genes and the translation and degradation of all 452 messenger RNAs. And so we don't have time to discuss the exact way we do the parameterization today, but we actually influence our well-stirred model using the statistics and results of this spatial model. So we can predict the number of active degradazomes, active RNA polymerases, and active ribosomes as functions of time, where this initial transience you're seeing here is a result of the fact that we initialized each of these complexes inactive. So no ribosomes were reading any messengers, no RNA polymerases were reading any genes. And then, as I mentioned, we also can predict the full distribution of the mRNA half-lives in Syn3A. And so here we got an average of about 2 minutes, a 2-minute half-life, and median of about 1.5 minutes. And then this long tail that's out to 15 minutes has actually been observed in Bacillus subtilis, and the average and median are in agreement with average measured in Mycoplasma gallisepticum. And so when we move on to the well-stirred model and go further than 20 minutes into a cell cycle, we can actually get to DNA replication. And so we were helped by Hamilton Smith with this mechanism. And so effectively, the initiating protein DNA has a high affinity signature, which is highlighted in red, where its domain 4 will bind to these 9 base pairs. It is flanked by 2 low affinity signatures, or 7 out of 9 matches. And then those neighbor a 91 base pair AT-rich region, to which the domain 3 of DNA can bind to single-stranded DNA. So the general mechanism is you will bind those 3 sites near the origin. They cause a little bit of torsional strain on your double-stranded DNA to expose the AT-rich region as single-stranded DNA, to which you will have polymerization reactions occur, and you will form a DNA A filament stochastically with on and off rates that were measured using SM-FRET. Then once you have formed, based on the length of that AT-rich region, we assume it takes 30 DNA A, and then you can load your riposome machinery and begin DNA replication. And so just some results of this, I have 2 representative cells on the left where we allow for an initial replication initiation event, and those are in the red and blue on the left. And then after you see that then there is no behavior for about 40 minutes. That's the period during which the DNA is being replicated. And then late in the cell cycle, you see the cell in the red shades has 2 more replication initiation events occur, 1 for each daughter chromosome. Whereas the blue shades, there are just fluctuations and no DNA A filament of length 30 is made. And so that depends on the stochastic reactions, the stochastic gene expression reactions forming DNA A. So the first cell generated enough DNA A by the end of its cell cycle to have 2 more replication initiation events, whereas the blue cell did not. And then the full distribution of these original replication events and the daughter replication initiation events is shown on the right. And so then we can get an idea of the number of chromosomes in a cell. The idea of multiple replication initiation events was actually influenced by qPCR experiments done by Andres Cook at the JCVI. So on the right, we have log phase and stationary phase ratios of origins and quarter to termini. So quarter, we just mean 3 o'clock on the chromosome. And so as you can see, both of them, there's more than 3 times as many origins as termini and more than twice as many quarters. So there certainly has to be more than one replication initiation event per cell cycle to achieve these ratios. And so our results then, on the left, we initialize each cell with 1 chromosome at the moment. That's our initial condition. And as you can see, by around 60 minutes, you will achieve 2 copies of the chromosome, where the solid line is the average, and then the shaded region is the full range of the whole population. And then at the end of the cell cycle, you get some cells where you have no further replication events, with the plateau at 2, up to you almost complete the replication events for the 2 daughter chromosomes by the end of the cell cycle for some cells. And so the result of this also...
We need to wrap up.
All right. So I have... so generally this affects nucleotide pools, so multiple replication initiation cells. You get you get a notion of homeostasis with nucleotide pools, where the pools are much more steady as you include more DNTPs into DNA. We also get pretty good protein homeostasis, where the average concentration of proteins are rather steady throughout the cell cycle, which means if you were to double the count of proteins, as this distribution on the right shows, you would double the counts of proteins as the volume is doubled. And lastly, we calculate ATP costs over the whole cell cycle for every single reaction in the cell. And most notably, we get the total cost of transport in this cell. Importantly, Syn3A has very few synthesis pathways and has to uptake most of its nutrients. So it turns out that actually one of the highest ATP costs in this cell, being anywhere from 10 to 20% of its total ATP cost, is just uptaking nutrients from its environment. And so just a few highlights of rules of life for the cell to conclude. So multiple DNA replication events are essential to maintaining and controlling the metabolite concentrations and giving some degree of homeostasis. With little to no regulation, the stochastic fluctuations can lead to a lot of undesirable behavior in Syn3A, but in general, it requires a lot of ATP to uptake its nutrients, and those need to be maintained well. And lastly, mRNA half-lives depend on the number and accessibility of active degradasomes and your particular DNA configuration in that particular cell. I would like to thank everyone listed here, the JCVI, for hosting, and if I have time for any questions, I will take them.
Well, dazzling, Zane. Questions, anyone?
I have a question, if I can ask.
Yes, please do.
Okay, yeah, also my video starts. Hi, I'm Lorenzo. I'm from Wageningen University, and I was actually very intrigued by the discovery that also mycoplasma undergoes multiple replication events per cell cycle, apparently, because I'm just approaching the mycoplasma field. I'm more, let's say, I work more in coli, and I know that in that organism, the degree of overlapping replication is, let's say, in accord with the growth conditions that the cell grows in. So I was wondering whether there are different media which promotes different growth rates of mycoplasma species, and whether the DNA copy number has been observed in different growth conditions to see whether when the cell grows more happily, let's say, then we observe also a higher number of copies of the DNA.
So my quick answer to that is, I unfortunately am not a seasoned mycoplasmologist, but my notion with Syn3A is that I'm not shocked, because there is, as I mentioned, there's little to no regulation left, including that for replication initiation. The only mechanism being present being that DNA A mechanism. So our thought is, as long as you have the DNA A protein available and you've synthesized enough of it, then it will find origins and it will start replication initiation events. But on the measurements, I don't know. Perhaps not to direct it to John Glass, but he would be. Do you know anything, John?
No, I'm not. I can't help you with that.
That's fine.
So if I
We can work on it. Lorenzo, get in touch with me.
Yeah, I will, thanks. But if I understand correctly, like, so your hypothesis on this is basically just a lack of regulation due to the minimalization of the genome of Syn3A, correct?
The genome is minimalized, yes, and therefore the controller of replication initiation in Syn3A is the initiator protein DNA-A.
Yeah.
And so as long as it's there and it has origins to find, it'll find them.
Yeah, definitely. Okay, thank you very much. And I will get in touch with you, John.
John, this is Bernard Paulson. Can I ask a quick question?
Sure.
Oh, yes.
So I'd like to ask if this model builds up significant electrical gradients and whether electron diffusion is accounted for in that master equation, and also whether you balance the osmotic pressure across the cell membrane.
Oh, that's a good question. So generally, the answer is so. So we don't do anything with osmotic pressure. We assume that the cell is perfectly spherical at the moment, and don't treat anything in terms of any of those forces. However, with the master equation, whenever we moved to the well-stirred system, where we assume the whole cell is well-stirred, we actually have to incorporate some probabilistic factors, and that's what I mentioned. We use the results of the spatial model to influence the well-stirred model to give us a notion of how probable is it for an active degradosome to find a particular messenger RNA? How likely is it for an active RNA polymerase to find a particular gene, and that sort of thing?
Yeah, I just asked this question for historical reasons. It's been over 30 years ago now, but the first whole cell model was built for the red cell, where many of these processes don't matter. But the most difficult part of that model was to balance the osmotic pressure and to maintain electron neutrality across the cell. And there's also a consideration that needs to eventually be looked at, which is electron diffusion, which sometimes is stronger than molecular diffusion. And those processes are not trivial to include, but something that you should probably think about going forward.
Yes, in the metabolic reactions, the metabolic reactions are actually charge balanced and proton balanced, but we haven't
Right.
got those incorporated in a sophisticated manner into this part of them.
Yeah, it would be the sodium, potassium, and chloride that are the big players in these equations, and something to consider going forward, like I said. So way back in the first model, these things were actually quite dominant, as it turns out, when you started doing the computation. So there will be some exciting times ahead to put these in there. And when it comes to rules of life, in terms of charged membranes and stuff like that, you're going to see these phenomena show up as being significant.
Oh, certainly. Zen and I have had a couple discussions already of hypotheses on how to go about including this sort of thing, but it was already an immense amount of work to even get to this point. So that is a next stepping stone that we are definitely strongly thinking about.
Thank you.
Yeah, thank you for the question.